Paramecium is a ciliated unicellular protoctist. The cilia are similar in structure to those found in the trachea of a human. The cilia beat to move paramecium through the water in which it lives. Figure 6.1 shows paramecium. Paramecium has anterior and posterior ends. Generally, the cilia beat so that the organism is moved forwards. Sometimes reverse movement is needed, for example, when the paramecium meets an obstacle. The direction of the beating of the cilia is linked to the difference in concentration of calcium ions inside and outside of the cell. There is usually a higher concentration of calcium ions outside than inside the cell. When the paramecium touches an object, its cell surface membrane becomes deformed. The membrane potential becomes more positive inside the cell. The organism moves backwards for a short time. Suggest the sequence of events that occurs to cause the paramecium to move backwards when it touches an object. This question is really easy since we can just answer using these bullet points. First of all, here it is said that the direction of beating of the cilia is linked to difference in concentration of calcium ions inside and outside the cell. Normally, there is a higher concentration of calcium ions outside than inside the cell. So obviously, the calcium ions will diffuse inside the cell, and this will also happen because when the paramecium touches an object, its cell surface membrane becomes deformed. So when this happens, the calcium ion channels open, or you can say, let's start it like this. Right? And then obviously, since the concentration of calcium ions is higher outside the cell, so they will diffuse down the concentration gradient. And then it is written here that after this happens, the membrane potential becomes more positive inside the cell, and then organisms move backwards. This is how cilia will beat. Suggest how paramecium ensures that there is usually a higher concentration of calcium ions in the surrounding water than inside the cell. Okay, this is hinting towards active transport, because we know that from the above bullet points that usually there is a higher concentration of calcium ions outside than inside the cell. So to maintain that concentration outside, paramecium must perform active transport of calcium ions against concentration using ATP. So active transport Okay. Paramecium has a contractile vacuole that fills up with water. When it is full, the contractile vacuole contracts to expel the water. The rate of contraction of the vacuole depends on the water potential of the surrounding water. Name the process by which water enters paramecium. That's osmosis. Suggest the relationship between the rate of contraction of the contractile vacuole and the water potential of the surrounding water. Obviously, if the rate of water potential is higher in the surrounding water, the vacuole will fill up faster with water and it will get full faster, and then it will contract to expel the water at a faster rate. So the higher the water potential, the faster the rate of the contraction. Okay. Describe how the DNA of paramecium differs from that of a prokaryotic cell. This is actually quite an easy question. You just have to memorize that the DNA of paramecium is first of all linear. And remember, they are asking you to compare with the prokaryotic cell's DNA. And it is associated with histones, or you can even say with protein, and prokaryotic cells' DNA is not associated with histones. And also the biggest difference, it is contained in nucleus, or you can say it is surrounded by a nuclear envelope, while that is not the case in prokaryotic cell. They are naked circular DNA in prokaryotic cell. Question 7. Figure 7.1 is a diagram of a section through a mitochondrion. Using figure 7.1, state the letter which indicates the site of the Krebs cycle. Site of the Krebs cycle is mitochondrial matrix, and that would be S. Oxidative phosphorylation takes place in inner membrane of the mitochondria, so that's R. And decarboxylation will also be S because it happens in the Krebs cycle. Suggest one function for the circular DNA in figure 7.1. Circular DNA. So since DNA consists of genes, so those genes will code for mitochondrial proteins and enzymes that are involved in the function of the mitochondria. So remember that transcription takes place due to DNA. During respiration, exchange of substances takes place between the cytoplasm and the mitochondria. Complete the table below to list 3 substances that enter the mitochondria and 3 substances that leave the mitochondria. First of all, entering the mitochondria would be oxygen, because we know that oxidative phosphorylation takes place. So, oxygen. Second thing could be definitely pyruvate, because it is needed for the link reaction, which is also taking place inside the mitochondria. So, pyruvate. And then you can write ATP or phosphate, or you can even write fatty acids. We know that ATP or phosphates will be used by the ATP synthase during chemiosmosis, and ATP synthase uses ATP and inorganic phosphate to make ATP. So we can write ATP. Substances that leave the mitochondria: carbon dioxide. Carbon dioxide is released in Krebs cycle in link reaction, so that ATP would leave mitochondria. That is released in electron transfer chain. Water, which is made as the last product in electron transfer chain. The poison cyanide binds with cytochrome oxidase, one of the carriers in the electron transport system. Suggest how ingestion of cyanide by humans leads to death by muscle failure. Obviously, if this poison is binding with one of the carriers in the electron transport chain, it would stop the electron transport chain. And if ETC is stopped, then Krebs cycle or link reaction would also stop, because there would be no NAD and FAD to take up hydrogen. Let's write that down. You can also say that the proton gradient would not be set up for chemiosmosis, and there would be no proton flow through ATP synthase, and then ATP would not be produced. And why is the proton gradient not being set up? Because if the ETC is stopped, there would be no energy to pump the hydrogen ions to set up the gradient. Suggest how ingestion of cyanide by humans leads to death by muscle failure. Okay, we also need to write how will this lead to muscle failure. We know that ATP is required to help the sarcomere contract, right? As there is ATP in the myosin head, which helps the contraction take place. So you can write that the most important organ in the body is obviously heart. If the the muscle of the heart doesn't contract, the heart won't work and person will die eventually. So you can say that heart muscle tripalmitin is a triglyceride. The chemical equation for the aerobic respiration of tripalmitin is, okay, so aerobic respiration. Calculate the RQ value for tripalmitin. Give your answer to 2 decimal places. Show your working. The formula for RQ value: volume of carbon dioxide released, volume of oxygen taken. Released is 1 or 2, and the volume of oxygen taken is 105. That's 0.70. Explain why the usual RQ value for respiration in humans is between 0.7 and 1. So since they're saying that it is still 1, so definitely anaerobic respiration is not being considered here. So respiration is done aerobically. For anaerobic, it's greater than 1. And since it's between 0.7 to 1, there must be a mixture of substrates being respired. Also, since humans have a lot of different tissues, so different tissues respire different substrates. That's also one of the reasons why the RQ value is between that. Question number 8. In a species of snail, shell color is controlled by a gene with 3 alleles. Allele CB codes for a brown shell. Okay. Allele this codes for a pink shell. And allele CY codes for a yellow shell. Okay. Allele CB is dominant to both. Okay. CB and CY. Okay. Allele CB is dominant to CY. Okay. The shells of the snail may be banded. They have dark strips, or non-banded. The allele for non-banded is dominant to allele for banded. State what is meant by the terms dominant and allele. These terms are very important and they are asked in the papers quite a lot, so make sure that you memorize these. So dominant is the allele that is expressed in the homozygotic and heterozygotic conditions in both of them. So an allele Allele is one of the 2 or more alternative nucleotide sequences at a single gene locus, or you can simply say variant forms of a gene that is expressed in different forms. So one of the forms is called an allele. A cross between a brown non-banded snail and a pink non-banded snail produces some offspring that are both yellow and banded. State the genotypes of both parents, brown and non-banded. Okay, so since they have said that the offspring are both yellow and banded, which means one of the allele of the yellow color should be present. So it should be CB, which is dominant over pink and yellow, and one should be CY because of this yellow. And for the parent is non-banded, so capital N is for non-banded, which is dominant over this small n, which is banded, and we know that offspring is banded. That's why we are taking the heterozygous condition and not homozygous and n. Okay, and similarly for pink, you take this pinks, it's CP. Okay, CP, CY, and we know that CP is dominant over CY, and it is also non-banded, so NN. List the parental gametes. Okay. So I hope you know how to make gametes. So to make it easier, I'll do it in a different color. Simply, it's very easy to make gametes. What you have to do is just, first of all, take this one, CB. Make the gamete with this N, capital one. So CB, N. Okay, that's one gamete. The next one you're going to take this CY. Or you can just take the CB again with this small n. CB, small n. This is the second gamete. Now, shift on to CY. Take CY and capital N. So CY, capital N. That's the third, and CY and small n. So CY and small n. These are the 4 gametes. And repeating the process for this one as well, you will have CB, capital N, CB, small n, CY, capital N, CY, small n. state the genotype of the offspring that are both yellow and banded. Since we know that the allele for brown color and for pink color is dominant over the yellow color, so it should be a homozygous condition for the yellow color to express. So it would be CY, and for banded, again, the non-banded was dominant, so the condition must be homozygous for the offspring to be banded. So does the proportion of offspring expected to be both yellow and banded. Since we know that the expected ratio of a dihybrid cross is 9:3:3:1, where this is the homozygous for both of the genes. So since both of these genes are homozygous recessive, so these are coding for this ratio. So the total is 16. So our proportion would be 1:1/16, right?